We come to the next presentation, to the visual cortex as an example of anatomical functional integration. It's Matthew Glasser from Washington State University, St. Louis. Matthew.
Thank you. So I'll be telling you about myeloarchitecture and probability maps and retinotopy of the visual cortex. So first, in vivo cortical architecture, myelin maps and gradients as areal boundaries, producing precise probabilistic maps by using processing methods that allow for more accurate spatial localization, and then finally, retinotopic organization of the visual cortex revealed with fMRI and resting state functional connectivity. So first, in vivo architecture. So very briefly, a historical perspective on myelin mapping of the cerebral cortex. It actually goes back to the Voghts, Oscar and Cecile Voght, who were among the first brain parcellators. And they recognized distinct cortical areas based on differences in several myeloarchitectonic parameters, including the overall myelin content, which would be sort of like the average of this entire histological slice here, the number and strength of the tangential fiber bands, the bands of Baillargeon, and the density of radial fibers, which are these that are coming up from the white matter towards the peel surface. And the Voghts thought that each cortical hemisphere contains around 200 myeloarchitecturally distinct cortical areas, which, as may have been said earlier by David, is based on what we know between monkeys and humans, it seems about right. So how do we do myelin mapping in vivo? What are the MR contrast mechanisms? So here we have an EM of a myelinated axon. You see the wrapping of myelin there. And there are several properties here that make it visible to MRI. It's rich in lipids. It's co-localized with iron, particularly within the cortical gray matter. And it restricts the motion of some nearby water molecules. And so these properties lead to several forms of MR contrast, including T1 contrast, T2 star contrast, and magnetization transfer. And each of these contrast mechanisms has been verified by taking a brain, scanning it, and then sectioning it and staining it for myelin and seeing that the slices, similar slices histologically in MRI are showing the same patterns. And those are some examples of studies that have done that. So the approach we use in the HCP is T1 over T2 cortical myelin mapping. So we use a T1 weighted MP range and a T2 space sequence. And these images contain all three forms of myelin, MRI myelin contrast. So myelin is bright in the T1 weighted image, so obviously the white matter you can see is very bright. The green outline here is the white matter surface, and then CSF is very dark, and the peel surface is the blue outline, so this is the central sulcus right here in this volume slice. If you look along the gray matter ribbon, you can see differences in intensity as you go along the gray matter ribbon. The anterior bank of the central sulcus here is higher, it's brighter than the intensity up here in the frontal cortex or down there in parietal cortex. But if we look in a T2 weighted image, we have the reverse pattern. So now myelin is dark, and we can see in the gray matter where it's darker here in the anterior bank of the central sulcus than it is in frontal cortex. And so because contrast is inverted between the T1 and T2, we can divide the images, which enhances our contrast while attenuating some of the MR bias fields. And even in these images, we can see myeloarchitecture, or we can see cortical areas rather. For example, area 4 here is a very thick, heavily myelinated area. We see nice distinct boundaries. Area 6, premotor cortex, is in front of that. Here in the fundus is area 3A. 3B here is also heavily myelinated, but you can see it's much thinner. And then area 1 is behind that. So it really helps us to visualize and compare across subjects to map this volume data onto the surface, which we've done here. So this white line here is basically where this volume slice intersects the surface. Typically, the most reliable measure that we use, at least in our data, which is 0.7 millimeter isotropic, is just averaging across all of the layers. At 0.7, we don't yet have enough resolution to be able to reliably see myeloarchitectural features though maybe we've got a hint of one here, for example. So there's some hints of it, but we need to be higher than 0.7 to really do that. So what do the myelin maps of an individual human connectome project subject look like? Well, we can see many cortical areal features are visible, including early sensory motor cortex, early visual cortex, early auditory cortex, the MT plus complex, retrosplenial cortex here is very distinct. This is actually a white matter pathway that goes through the gray matter, the perforant path, and the subiculum. Frontal eye fields. And we've got a hot spot here in the IPS, we think is the homologue of LIP and VIP. We also have very lightly myelinated areas that we can see, including the anterior insula, anterior cingulate, and temporal pole. So how have we validated these maps? Well, here we've got group average myelin maps, again, the central sulcus here, and we're interested in, if we wanted to find the boundaries of cortical areas, we're interested in where does the myelin content change most dramatically. So we can take the first spatial derivative, or the gradient, of this map here, which produces this myelin gradient here. And now the reds and greens and yellows are all where the myelin content here is changing rapidly. So we can put squares here along these gradients, and these squares are going to be in the same places across all the panels. And then we can compare these the myelin maps and gradients to probabilistic areas from cytoarchitecture. So these areas were mapped onto the individual subject surfaces and then registered on the surface, just like the myelin maps were, so it's the same surface registration algorithm being used here. And we can take a gradient of the probability map, just like we can take a gradient of the myelin map here, and we can see these gradients are very nicely co-localized. So for example, this bright stripe here of myelin area 4 very nicely co-localizes with the probabilistic map. 3A in between the two heavily myelinated regions is also co-localized, 3B, and so on. And we can see this in the right hemisphere just as nicely as the left. So to summarize this section, cortical myelin content can be measured with MRI in living brains. In vivo myelin content boundaries agree with postmortem cytoarchitecture. And early sensory motor areas tend to have more myelin, whereas many of the higher cognitive areas tend to have less. And gradients reveal the most likely location of areal boundaries. So next, on to making really good probabilistic maps. So typically what's done in neuroimaging is a volume-based analysis for comparing across subjects or studies. So the traditional approach is to align subjects with volume registration of a T1 weighted image to a group template. People often do a lot of spatial smoothing of the data in an attempt to correct for residual misalignments and other reasons. And they often summarize their group data for comparison across studies using 3D standard space coordinates or sometimes volume-based probabilistic maps. But there are some limitations here. We have relatively poor cross-subject and cross-study alignment of cortical areas, and blurring from just the substantial intersubject variability of folding patterns, the variability of areas to folds, and smoothing also mixes signals from different tissue types and across different brain areas. So the results of substantial uncertainty in spatial localization, basically being able to answer the question, do two studies find the same area with the same boundaries or not? And here's an example of this kind of uncertainty, which can be very frustrating. So we were interested in this region of higher myelination here outlined by the white squares, and we wanted to know what is the function of this area. Have people mapped it in functional imaging studies? And so we tried plotting the stereotactic coordinates of a variety of studies that looked at saccade tasks. And you see they sort of cluster around this region, but we don't have a clear agreement of boundaries between these studies and this region. So it's hard to say definitively that we've got the same area. So we can do better than this, though. So data from the sheet-like cerebral cortex is better analyzed and visualized on surface models. An early application of this to fMRI was Serino's retinotopic study here. So they've measured polar angle and eccentricity in the visual cortex. And this is one of the first that I'm aware of surface-based fMRI studies. And studies of the visual system have largely followed Serino et al.'s lead in analyzing and visualizing data on the cortical surface. So if you look at a retinotopy study, you're almost invariably going to be looking at surface representations. And as a result, we have a better understanding of the boundaries and fine details within the brain's visual areas relative to some of the other brain areas. So one of the keys here is surface-based registration, which substantially improves spatial localization in cerebral cortex. So it's just a fundamentally easier problem to align cortical areas in 2D on the surface than it is to try to align both the cortical areas and the folding pattern itself in a 3D volumetric registration. And it's easier to preserve the spatial relationships and borders between cortical areas on the surface. And area 2, which is always located on the anterior bank of the postcentral sulcus, is a nice example of that. If we take the histological definition of area 2 from 10 subjects and we register it in the volume, we get a probability cloud that looks like this. If we then map it onto the surface, we can see probability that's bleeding to a variety of places that we don't expect area 2 to be, like the anterior bank of the central sulcus or the IPS. But if we just make one change, we take the same cytoarchitectonic definitions and we register them, we map them onto the individual subject surface and register them on the surface, we get a much cleaner probability map with higher overlap. But often cortical folding-based surface alignment is blurry. So registration using folding of patterns alone often doesn't accurately align cortical areas across subjects in many parts of the cortex. Generally, if folding patterns are consistent across subjects and the areas are consistently located relative to the folds, like we saw in the last talk, those areas may be well aligned by folding-based registration. Another couple of examples are V1 and the early somatosensory and motor areas. But many other regions may still have significant misalignment after folding-based registration. Around MT is a challenging, more challenging one. We only get about 50% overlap there. In the IPS is another challenge, or the frontal eye fields. And here we have even some areas that are completely misaligned. So how do we do better? So we can align subjects based on features that are more closely tied to their cortical areas than is folding, such as the myelin maps, resting state functional connectivity, topographic maps, or even the cortical areal definitions themselves. And so new surface registration algorithms like multimodal surface matching written by Emma Robinson offer improved areal alignment with what I'm calling cortical areal feature-based surface registration. So let's take though we do start with folds as our initialization. So let's take an example of this with myelin maps. So as I said, the myelin maps are more closely tied to cortical areas than our folding patterns. This is a folding-based registration of myelin maps here. And we can see some areas are blurry on group average myelin maps registered with folding-based registration. So as we're going to see in a second, all of these areas that are outlined by white dots are going to get sharper. And we can see why they're a bit blurry. This is an individual subject here, and we've got misalignment between the areas, the white dots and the individual subject areas. But MSM improves the alignment of myelin maps, so now we get much sharper hotspots of myelin, and that's because the individual subject data is now better aligned. Resting state networks can also be used with MSM for cross-subject registration. So here we're going to have folding-based alignment on the top and resting state network-based alignment on the bottom here. So resting state networks have contrast over more of the brain than do myelin maps. So we've got these big oceans of blue here in cognitive regions where we don't have so much contrast in myelin content. We've got a lot more contrast in resting state networks. So we get improvement in alignment of functional connectivity, which is expected because that's the modality we drove the alignment with. But they still actually do a good job of aligning myelin maps. So there's common information between the myelin maps and the resting state networks that we're tapping into here. And I'm not going to show it for reasons of time, but task fMRI maps that are aligned with resting state networks also show improvement. We can even register binary cortical ROIs with MSM. So we're going to consider an 18-area retinotopic fMRI parcellation of 12 individuals by Guy Arbon's group here. And we're going to look at both the probabilistic maps and the maximum probability maps across the two techniques. So here are these retinotopic areas. So first, the probability maps. We've got folding-based alignment along the top and alignment driven based on the retinotopic areas along the bottom. And you'll see basically in every case we do better in terms of the overlap of the areas across subjects. In the maximum probability maps, we're going to focus in here around the MT plus complex. Here's the maps we get with folding-based alignment, which have kind of are jagged, and even some of the areas are hard to see. But if we use the retinotopic areas to drive the alignment, we now preserve the topology. Basically, we've got our nice cluster of 4 MT plus areas and our cluster of PIT areas here. That's much better preserved after using the aerial definitions to drive the alignment than the folds. Another issue we have to worry about with registration is group registration drift, and that's simply a mismatch between the group average data or the group average template and a typical individual subject. These are HCP data here, and these are just simply brain volumes as an example of where this is a particular problem. If you register brains to M&I space, one of the things you want is to reduce the variability in the brain size and shape, which we see here. The variability is being reduced. But if you also get this increase in brain volume, and that's not something you were after. You were just wanting to reduce variability. You didn't want to actually change what the average was. And it increases by 37%. And that's because when M&I space was created using iterations of registration and averaging, basically, the template drifted to a 37% larger brain size than the individual subject's, typical individual subject's brain volume. So these are the same brains here, it's just before and after registration to M&I space. And this is now baked into the standard space. If we fix this, then we wouldn't be in an M&I space anymore. So it's hard to fix, but on the surface, we can try to avoid making this mistake. And we can remove group drift by computing the group average registration and concatenating its inverse onto each individual subject's registration. So here's an illustration of how this effect can happen on the surface. So we're going to focus in here. So this is retinotopic areas and HCP myelin maps. Both have been registered with MSM, but using different information. So the retinotopic areas were registered using the aerial definitions, and the myelin was registered using the resting state networks. We can see reasonable agreement in this MT plus complex area in the left and right hemispheres, but you also see it's a little bit asymmetric. But if we remove this drift, we're focused there. If we remove this drift, we see that the alignment actually gets better in both hemispheres and more symmetric. So I'll try to flip between them a few times so you can see that. So if we do the aerial feature-based registration and we remove group registration drift, we can do really precise cross-study, cross-modal boundary comparisons. Here we're going to consider resting state functional connectivity and myelin maps. We're going to compare them to these retinotopic parcels here. So here we've got our seed for our functional connectivity, and we're going to focus in on the MT plus complex again. We've got stronger functional connectivity to the IPS in the red area and dark red area, MST and FST, but less connectivity between that seed and MT and V4T. However, if we put a seed in a different location, we see the opposite pattern. So what's really cool is this is one of those locations I showed you earlier that's pretty hard to align across subjects, but we've actually got this really nice agreement now between the boundary and functional connectivity and the boundary in the retinotopic areas. And we see that as well in the right hemisphere. Cortical data are best analyzed, registered, and visualized on surface models. Aerial feature-based registration improves the alignment of cortical areas over more typical folding-based registration. Areas and folds often don't agree. Sometimes they do, though. Group average registration drift makes cross-study comparisons less accurate, so it's worth removing it. And if we do our processing and alignment carefully, we can make very precise cross-study aerial boundary comparisons. So now the last topic, retinotopic organization of the visual cortex, and we'll talk a little bit at both with task fMRI, retinotopic tasks, and resting state functional connectivity. So how do we measure topographic organization in cortical areas? Unlike the sharp gradients that I was showing you in the myelin that form areal boundaries, topographic connectivity gradients tend to be smoothly varying and occur inside cortical areas. And many describe spaces outside of the body. For example, the visual space, the visual field, which we're going to schematically represent over here, is represented spatially in primary visual cortex. This is a left visual cortex here. So if we put the meridians on, so the right horizontal meridian is in purple here, and we see that has a particular location in primary visual cortex. Similarly, the vertical meridians have specific locations as well. Different parts of how close to the fovea or the periphery you are, the eccentricity, also have specific locations in visual cortex, foveal being on this side and peripheral on that side. So if we stimulate a specific part of the visual field, this leads to neuronal activity in specific parts of visual cortical areas. And that's because the corresponding parts of these areas are strongly connected. So if we stimulate here in the visual field, we'll get in particular activity there. Similarly, if we stimulate in a couple of other locations, we'll get specific activity there as well. And so we can measure this topographic organization using either a task paradigm or connectivity. So how do we get aerial borders out of topographic maps? Well, it turns out that polar angle reversals define boundaries between visual areas. So we've got our horizontal meridian in pink here, and our vertical meridians in cyan and yellow. And the vertical meridians define the boundary between V1 and V2, which we can see very nicely there. The horizontal meridian between V2 and V3, and this pattern continues further, as I'll show you in a second. Also, visual areas tend to have both central and peripheral eccentricity representations, and that information can be useful sometimes for parcellation as well. So many groups have done really beautiful work with retinotopic fMRI. Here's one of those examples where they took the polar angle reversals and they went all the way up into the superior parietal cortex, identifying cortical areas based on these reversals. Others have done, I've already shown you the Orban retinotopic maximum probability maps, but another study that recently came out actually found even more cortical areas out of Sabine Kastner's group. In this one, though, they used only the folding-based registration. They didn't use aerial feature registration, and you can see some on the edges there. The maximum probability map is kind of petering out. So it'd be really interesting to see this map based on aerial feature alignment. So what about using resting state functional connectivity for doing visual parcellation? So first we need to, since we don't have a task, we can't stimulate the retina here, we have to define our coordinate space in V1 instead of on the retina. So we've got to identify V1. We've got a nice gradient, but potential gradient here for V1 in the myelin maps. And it turns out if we seed the lateral geniculate nucleus, the thalamic relay station of visual information with functional connectivity, we get a very nice gradient that agrees with our myelin gradient. So we can identify V1 in this blue outline here. There are also a variety of other modalities, including the whole brain resting state gradients and task fMRI that show boundaries in this same location for V1. So we're going to define a coordinate space in V1 and generate visuotopic spatial regressors. We'll have our schematic up here, so our pluses mean a positive 1 and our minuses mean a negative 1 here. So we're going to generate some spatial regressors. Here we're contrasting the lower vertical meridian up here with the upper vertical meridian, and then we have the horizontal meridian is at 0. Similarly, we can contrast the left and right horizontal meridians. Both horizontal meridians versus both vertical meridians we can contrast, and we can also contrast foveal versus peripheral eccentricity. So we've got a few other regressors here. And one thing to keep in mind, again, there's a nonlinear transformation between retina space and V1 space. So we're linear in V1 here since we don't have a task. So we can generate whole brain spatial maps based on these V1 limited spatial regressors. How do we do it? Well, we do a weighted dual regression approach, and here we're just weighting by the vertex areas to make sure each square millimeter of cortex contributes equally to the regression. So for those of you not familiar with dual regression, one first does a spatial multimodal regression. You have all of your regressors and you regress them spatially into either group or individual subject time series. And then you generate a time course for each spatial map as the output of that. And then you take that time course for each spatial map, and again, you multiple regress them into the resting state time series, and you get new spatial maps. So while we were first limited to being only in V1, once we've been through this process, we have whole brain maps here. Although these still are biased a little bit by some other resting state signals. So we'll do a little bit more processing, and we're going to focus on this horizontal meridian versus vertical meridian contrast here. So how do we get some areal boundaries out of this data? We've got, so here's our contrast we're starting with. We're going to draw a line from the middle of V1 along the horizontal meridian up the visual areas this way. And this is the value basically from this map plotted over here. So you can look at both things here. So we can take the gradient of this map here, and now we're going to focus on more where does the polar angle reverse, which is basically minima in this gradient instead of maxima. So we can see a variety of these minima as we move up this way, and are indicated on the graph by the stars. And if we want to actually get a measure that's going to be the same within the cortical areas, we can focus not on the gradient magnitude, but on the vector. So basically, the vector is either going to be pointing towards V1 or away from V1, and it's going to reverse at the areal boundary. And so we can take the dot product of that vector with a gradient that always points towards V1. And so when we're pointing towards V1, we're going to get a positive value, away from V1, a negative value. And so now we get inside of V2, we've got all negative dot products here. Then V3, we've got positive ones. V4, negative ones, and so on. So we can use this to define retinotopic areas. Basically, wherever the sign changes, we can define a boundary. It's a little bit analogous to a field sign analysis that some of you may be familiar with. And we can see that these retinotopic areas are all heavily myelinated here. So how does this compare with more typical methods for finding retinotopy, basically retinotopic task? Well, we'll use our data we already looked at, the Orban's 12-subject group average retinotopic parcellation, and we'll compare it with these resting state defined boundaries. So both data sets were registered, again, with MSM aerial feature-based registration and de-drifted, so we should expect very good agreement between the boundaries. Indeed, we see very good agreement between V1 to V2, V3, V3A, V7, and V4, really along the paracentral region. But one thing to keep in mind is retinotopic fMRI doesn't often stimulate the far periphery because it's very hard to actually, when you're in a confined scanner, it's hard to actually hit that part of the retina with your stimulus. So we're able to map with resting state the full extent of these retinotopic areas, whereas that's very challenging to do with retinotopic MRI. And similarly, in the fovea, it's challenging to map because you need people to be fixating on a crosshair and not moving their eyes, and that's very hard for them to do perfectly. So the resting state visotopic parcellation can map both of these far peripheral and foveal regions better. So to summarize retinotopic organization of the visual cortex, fMRI has been very successfully used to measure retinotopic organization and define visual areas. It's also possible to do this with resting state functional connectivity. And the technique has a few advantages. It doesn't require a specific retinotopic task, and it can map the entirety of visual areas. In retinotopic organization, retinotopically organized visual cortex is heavily myelinated. And I'll take any questions in the little time I have left.
No questions. Okay, then I have one short question. How do you define the borders of the myelo areas?
So there we're looking at where does the myelin content change most rapidly. So we look at you have, let's say, here's your area 4, and then your 3A is here. And so you look, where does that function change? Where does the derivative of the function highest? So that's basically how we do it.
Yeah, but this is, the slope of this change is different at the different borders, right?
Right. So the larger answer is it's good to have multiple modalities, and that's one of those things I didn't have time to talk about today. But that is the whole story. So we use multiple neuroimaging modalities to do that.
Okay. Thank you.